Welcome to NPTEL certification course on Pharmacognosy and Metabolic Engineering. This is lecture 33. So, where I will narrate the recent discovery of strychnine biosynthetic pathway, and this discovery was reported again in the famous journal Nature last year, that is 2022. So, let us go. So, the concept covered, simply I have written strychnine's biosynthesis deciphered. That means, how strychnine is biosynthesized in plants. So, let us go to the slide number 1. So, our topic of discussion is biosynthesis of strychnine. Now, what is strychnine? Strychnine is basically a poisonous alkaloid obtained from the plant called Strychnos nux vomica. I am sure most of you or almost all of you are aware about a popular homeopathic drug nux vomica. So, the nux vomica the homeopathic drug is basically obtained from a very diluted extract, either seed or root extract of Strychnos nux vomica plant. So, because of this poisonous nature of the alkaloid, that actually fascinated the scientist or inspired the scientist for the past 200 years to determine the complex skeleton of this molecule, and they reconstructed the molecule in the lab. So, that means the synthetic organic chemist partly made the skeletons in separate event, and then they finally joined and made the synthetic strychnine. But that is not commercially feasible if you want to make strychnine, because the organic synthesis reaction are so complex and expensive. And at the same time, it was not known that how in the nature plants synthesize strychnine. So, that remained a mystery until recent now. So, scientists have solved this using chemical logic, by using chemical logic and a study of messenger RNA of Strychnos nux vomica eventually led to the or helped to decipher the whole pathway of strychnine biosynthesis operating in plants. So, this discovery again published in the famous journal Nature in the year of 2022 by Sarah Connor's group. So, the volume is 607 and page number 617 to 622. So, the first author was Hong et al. And the group leader was again Sarah Connor. from this time she moved from John Ennis Center to the Max Planck Institute for Chemical Ecology in Jena, Germany. So, the paper basically the affiliation given to this institute now. So, we are going to see what is it. Okay. So, let us go to the okay. For that I will also tell few things, because strychnine is a bioactive molecule. Its derivatives could have pharmacologically useful properties without being poison. So, why this is important? Because once you know the biosynthesis of strychnine, once you know the enzymes and genes of the pathway, and once you can synthesize the strychnine in the laboratory, then it is possible to make derivatives of strychnine, and these some of these derivatives may not be poison, but have more pharmaceutical acceptance. So, knowing the biosynthetic route could help the chemist generating the strychnine scaffold, and possibly will be making in future such derivatives using the synthetic biology. And what is basically the ultimate rationale for doing this project? Okay. Now, let me go to the pathway. Okay. Now, another interesting point I tell before I go to the pathway, that once this paper was published, so several news agencies they took interview, and one of the interviews the Hong, who actually the senior postdoctoral researcher and the first author of this paper, what he said that they basically failed to produce strychnine because that was ending with the formation of pre-strychnine. So, I just write it here, pre-strychnine, P-R-E-S-T-R-Y-C-H-N-I-N-E. So, pre-strychnine is the penultimate molecule of the pathway, and from pre-strychnine, strychnine will be synthesized. And what I said, that this is a very toxic molecule. Okay. So, this conversion, that means the enzymes responsible for this conversion, they fail to really identify the enzymes, or they fail to convert pre-strychnine to strychnine. Finally, what they said, that when they left the pre-strychnine in the laboratory in one Friday, and by mistake or whatever may be, and when he came back in next Monday and started seeing that before throwing it out, that time they found that this pre-strychnine got converted into strychnine. That means, the some amount of pre-strychnine got converted into strychnine. That means, the solution containing the pre-strychnine, when they analyzed, they found that some portion of pre-strychnine got converted into strychnine. That means, from there it was clear to them that basically spontaneous conversion is taking place. Which may not be enzymatic. So, may not be enzyme, and eventually by doing several experiments, they indeed confirm that this last step is basically spontaneous in nature. So, with this now let us go to the next page. So, the biosynthesis. So, how I will tell this? First I will tell the basic biosynthesis what has been discovered, and then I will tell that how they have confirmed this biosynthesis using Nicotiana species. So, again this is actually the part of this indole alkaloids, because the intermediate we will start from gaseocyzine. So, let us start from there, gaseocyzine. Gaseocyzine will be converted to through a unstable intermediate, which is dihydro. Okay, so, do not worry student, you do not have to remember these difficult names, this is just for your information. And that makes norfluorocurarine, RA-rin. So, the steps so, the enzymes are SNV stands from strand for strychnose nux vomica, GO gaseocyzine oxidase. So, they have used another species. So, that they have mentioned in the paper, that is strychnine species. So, they put it in this way SP, and this they are also GO, they have also confirmed. Now, the second species they are that is not producing strychnine, but producing another compound. Okay. So, this is number 1, this is number 2, and then finally, from this by 2 reactions this norfluorocurarine is formed. So, therefore, I put 2 arrows here, and the enzymes are SNV NS1 and SNV NS2. That means norfluorocurarine synthase 1, norfluorocurarine synthase 2, and this in the publication they have marked as 4. I will simply follow their process. So, this in the next step will be converted into 18 hydroxy norfluorocurarine. So, this was marked as 5, and this in the next step will be converted into a compound called Wieland Gumlich aldehyde, and the enzyme is SNVWS, that is Wieland Gumlich synthase. So, this they have marked as number 6. This now this is one of the major intermediate. From there one pathway emerges, leading to the formation of a compound called diabolin. This they have given the name number 8, okay. And the route which moves towards formation of strychnine, that makes next product as pre-strychnine. Pre-strychnine they have marked as number 9, and pre-strychnine in the next step makes strychnine. This they have numbered as 10, and as I said, this one is pre-strychnine to strychnine. This is a spontaneous. So, it releases a molecule of carbon dioxide along with a molecule of water, and this produces strychnine along with isostrychnine. So, this number is 11. Now, the enzyme responsible for this reaction, that means Whelan-Gomlich-aldehyde towards pre-strychnine, that is SNV 80 acetyl transferase. So, next this strychnine will be converted into 10-hydroxystrychnine. And that in the next step will be converted into beta columbrin. Beta columbrin, and the enzyme responsible for this reaction is SNV 10H, that means 10 hydroxylase, and this one is SNV O methyl transferase, and the next one converts these to 11 dimethyl brucine, and that finally converted into brucine. So, brucine will give the name 15 and columbrin will give 13. Okay. Diabolin, I will use a different, I can use a different color, sorry. Diabolin. So, when I put diabolin, better I use, don't use this one. Okay. So, diabolin accumulates in another species, not in Strychnos naxvomica, but another Strychnos species, where strychnine is not produced, instead it produces diabolin. That means, there the pathway moves from Wieland-Gumlich aldehyde one step by a acetyl transferase, it makes diabolin, whereas in case of Strychnos naxvomica, the pathway moves in this direction towards the formation of strychnine and strychnine, and then subsequently it produces the other alkaloids. Now, the enzymes for these last steps I must put it, that is the last one, the brucine formation, this is also SNV O methyl transferase, and this one is basically SNV 11 hydroxylase. And what is brucine? Brucine is basically the dimethoxylated derivative of strychnine. So, brucine is basically the dimethoxylated derivative of strychnine. And it accumulated in the roots of Strychnos naxvomica, found in Strychnos naxvomica roots. So, this is in brief the biosynthetic pathway leading to the formation of strychnine, brucine and diabolin in the other species. So, they have used both the species for this study. So, basically they have done a very detailed transcriptome analysis from both the species, and then they did in depth gene annotation and subsequent cake pathway analysis and whatever analysis required, that is beyond the scope of discussion in this class. And from there that then leads to the discovery of these enzymes. Now, what I am straight away writing that SNV-GO, SNV-NS1, but what is important after this, that this needs to be demonstrated in another heterologous system to confirm its function. So, and just for your information that the gaseocyzine is basically it is coming from the indole alkaloid pathway, which started from GPP and tryptophan. If I say at that point, so GPP is basically the terpenoid root which ultimately makes ecologanin, and tryptophan is coming from the secumate root, and with that ultimately contributes towards the formation of strictocidin, and then gaseocyzine is formed, and then the reaction proceeds. Okay. Now, there are some interesting observations which I need to tell you. So, first of all, so let us go to the next slide. So, now the validation of the function. So, what we write that validation of these genes, or rather we should write validation of the functions of these genes. So, for that what they have done, they have used as I said a heterologous species, that means they have used Nicotiana benthiamiana, Nicotiana benthiamina. So, that means here they try to reconstitute the strychnine biosynthetic pathway. So, so they achieved what I should write it, they achieved transient expression of all enzymes, that is starting with SNV GO, SNV NS1, SNV NO, SNV WS, this is W please, WS, then SNV AT, then SNV 10H and SNV OMT. Okay. That means they have transformed Nicotiana benthiamiana plant with all the genes. That means the pathway will be operating. But now you should ask yourself whether the Nicotiana benthiamiana will produce the strychnine or not. What will be the answer? Yes or no? No, it will not produce, because Nicotiana benthiamiana will not get gaseocyzin. Okay, gaseocyzin is basically the product of the indole pathway, that is not operating. So, then what needs to be done? What needs to be done, that you have to take a transgenic leaf expressing Nicotiana benthiamiana and there you do infiltration of gaseocyzin ISSOSCHIZINE, and also you require malonate source and simply I write and malonic acid, that was supplied in the form of disodium malonate. So, when you infiltrate in a live plant, simply inject it and allow the plants to take it up. Then after 1 week you harvest and check for the alkaloid contents, and what you will find, or what they have found, is basically they found the presence of strychnine. So, I write it here, presence of strychnine, isostrychnine, then columbrin and brucin. So, they found this in the, so when you find this but you see I have kept a blank space here. Actually what happens that when they transferred all the genes, it did not produce the strychnine, columbrin, brucin products. So, what happened basically that the malonic acid what you are supplying. So, that malonic acid will joins with a CoA that may be and then it forms a malonyl CoA, and malonyl CoA should be acceptable to the SNV80. That means, the SNV80 which is the this step, So, is in order to have the SNV80 function, you need to have the malonyl CoA added in it. So, okay, then only it makes the pre-strychnine. Now, what happened that SNV80 when you put the SNV80 then SNV10H, that means the all the genes of the pathway, it is not producing the strychnine. If strychnine is not produced, it cannot make beta columbrin or brucin. So, then they found that basically the SNV80 is not getting the accession of the malonyl CoA. So, in order to get that, what they have done, they have transferred here a gene from Arabidopsis thaliana, so which is ATAAE, that is added here. So, ATAAE is basically acyl activating cytosolic enzyme. This is basically the acyl activating cytosolic enzyme that produces malonyl CoA accessible to cytosolic, that produces malonyl CoA accessible to cytosolic SNV80. Okay. So, that means ATAAE to be added here in the pathway, and then the pathway will function and eventually it makes the strychnine. Okay. So, basically some malonyl transfer is there, and this finally makes the thing. So, and therefore, now what we can say, the heterologously reconstituted pathway, I just write it here one sentence, the heterologously reconstituted pathway matches the physiologically relevant pathway in strychnose nux vomica plant. Okay. So, that means they have reconstituted the pathway and it basically produces the product. Now, so to make this story short, so what they have done, that initially they have not transferred all the genes together, they have done stepwise. So, they made it in two combination. In one combination they have infiltrated say SPGO, SPNS1, SPNO and SPWS. Why? That also I need to tell, because what I said that there is an equation, the strychnose species, other species which is producing diabolin, but not strychnine. For that, what is required, that if you go to the pathway, so suppose the pathway is meant for the formation of diabolin in the other species of strychnose, then what we need to show here is this, that SPGO, then SPNS1. So, there will be another SPNS1, then there may be SPNO, which I have not shown, okay, and then there will be SPWS, right. So, if you take these genes, right, and of course, this one, then what we will see that it produces the, or if you do not take up to this, if you take only up to this much, then what will happen, this basically leads to the formation of Wieland-Gumlich aldehyde. That means, expressing SPGO, SPNS1, SPNO and SPWS leads to the formation of Wieland-Gumlich aldehyde. So, first they have confirmed. So, in the another combination, what they have done, they have taken SPGO, SPNS2. So, that means what you can also write here by putting a slash here, that SPNS2. So, this one, for example, I will use another color, okay, color is not sufficient with this one. So, the second combination may be this, then this and this, and along with this, that leads to the formation of diabolin. Okay. So, in order to produce the like this way, they have checked the formation of this short pathway, which is operating in one strychnose species, and also they have checked the formation of Wieland-Gumlich aldehyde, which operates in both the species. And next what they have done, the next step, they have added all the genes what I have shown in the next slide, that all these, and eventually they express all these genes in Ecosia benthia manner. This leads to the deconstitution of the strychnine pathway, and still it did not produce strychnine unless you add a AAE, acyl activating cytosolic enzyme, that actually allows the malonyl CoA get accessible to SNV80. So, only that is only one enzyme you have to add from another source, mind it, because it is a Nicotiana system, it is not the strychnose. So, whatever you wanted to do, plant will not do. Okay. So, they are clever. So, you have to conquer. So, that is why they have conquered, and eventually they made the strychnine, and this strychnine, what they said, this heterologously reconstituted pathway matches the physiologically relevant pathway in strychnose nux vomica. So, this is basically all about it. What I thought that I will draw some structure, but I will keep it in for the next class. So, next class I will make a brief overview of whatever I have taught in this indole alkaloids, in all this terpenoid indole alkaloids along with the strychnine. So, this all I put a brief overview with slides and showing the, I will show you the plants how they look like, and then I will show you the schematic diagram with the enzymes and genes, and also I will show you some histochemical localization, some what people have done, as well as the immunochemical localization, immunohistological localization of these enzymes. All these things will come in the next class. With that I will end the indole alkaloids, and once I finish that, the subsequent class I will talk about another interesting alkaloid which was also discovered in 2022, the pathway which is called colchicine, and once I finish the colchicine, all alkaloid will be covered. So, with this I end this class. Thank you.